Genetic expression from the X chromosomes plays a significant role in shaping brain function because females carry two X chromosomes, while males carry one X and one Y. At first glance, it might seem like having two X chromosomes would simply double the expression of all X-linked genes, but the reality is more nuanced. The body has a mechanism called X inactivation, where one of the X chromosomes in each cell is largely silenced to prevent overexpression. However, this process is not complete, and that's where important differences begin to emerge. Some genes on the X chromosome escape inactivation, meaning they remain active on both copies. This results in higher levels of expression for certain proteins in females compared to males. Many of these genes are involved in brain development, synaptic function, and cellular maintenance. As a result, this difference in gene dosage can subtly influence how neurons grow, connect, and respond to signals. Another layer to this is that X inactivation is not uniform across all cells. In some cells, one X chromosome is active, while in others, the alternate X is active. This creates a mosaic pattern across tissues, including the brain. In practical terms, this means that female brains often have greater cellular diversity in terms of gene expression. Different populations of neurons may express slightly different versions of the same genes, which can influence how neural networks operate as a whole. This mosaicism may contribute to resilience in certain contexts. For example, if one version of a gene carries a mutation or is less efficient, other cells expressing the alternate version can compensate. This has been proposed as one reason why females may show some protection against certain genetic disorders that are linked to the X chromosome. At the same time, this variability can also introduce differences in how the brain processes information, particularly in areas related to cognition and emotional regulation. These genetic dynamics also interact with developmental timing. During early brain formation, gene expression patterns guide how neurons migrate, form connections, and establish circuits. Even small differences in expression levels can lead to lasting changes in how these circuits are organized. Over time, these initial patterns are further shaped by hormonal influences and environmental input, but the genetic foundation remains an important starting point. It's also important to recognize that genes do not operate in isolation. They influence the production of proteins, which in turn affect how cells communicate, how synapses strengthen or weaken, and how the brain adapts to experience. In females, the unique pattern of X-linked gene expression can impact processes like synaptic plasticity, immune responses in the brain, and the regulation of stress pathways. This creates a system where genetic variation is not just about presence or absence, but about degrees of expression and interaction. The result is a brain that may process signals with slightly different sensitivities or priorities, depending on how these genes are expressed across different regions. Over the lifespan, these genetic influences continue to interact with hormonal changes and lived experience, meaning their effects are not static. Instead, they provide a flexible framework that contributes to the overall dynamics of brain function in ways that are subtle, but mean early hormonal exposure during development plays a powerful role in shaping how the brain is organized, and much of this occurs before birth and in the early postnatal period. During these critical windows, hormones act as signals that guide how neural circuits are formed, strengthened, or pruned. These effects are long-lasting because they influence the basic architecture of the brain rather than just moment-to-moment activity. One of the key factors in this process is the presence or absence of a surge in testosterone. In typical male development, there is a significant increase in testosterone during prenatal life. Interestingly, much of this testosterone is converted into estrogen within the brain by an enzyme called aromatase. This locally produced estrogen then acts on neural tissue to shape the development of specific circuits, particularly those involved in reproductive behavior, aggression, and certain aspects of motivation. In females, this early testosterone surge does not occur at the same level. As a result, the brain develops along a different trajectory. It's not that structures are missing or added, but rather that the sensitivity, connectivity, and balance of neural circuits are tuned differently. These differences can influence how regions of the brain communicate with one another, including areas involved in emotional processing, memory, and decision-making. Timing is crucial in this process. There are specific periods during development when the brain is especially responsive to hormonal signals. If these signals occur within that window, they can have organizing effects that persist throughout life. Outside of those windows, the same hormones tend to have more temporary, activational effects rather than permanent structural influence. This distinction helps explain why early hormonal exposure has such a lasting impact compared to hormonal fluctuations later in life. Another important aspect is how these hormones influence gene expression. Hormones like testosterone and estrogen bind to receptors that act directly on DNA, turning certain genes on or off. This means that early hormonal exposure doesn't just affect brain structure at a macro level, it also changes which genes are active in specific cells, further shaping how those cells function and connect. These early hormonal effects also interact with other biological systems, such as the stress response and sensory processing. For example, differences in how the amygdala and hypothalamus develop can influence how individuals respond to threat, novelty, and social cues. These patterns are not rigid, but they do create tendencies that can be observed later in behavior and cognition. It's also important to understand that this process exists on a spectrum. Variations in hormone levels, receptor sensitivity, and timing can lead to a wide range of outcomes. This is why individual differences are so prominent, even within the same sex. Biology sets certain parameters, but it does not determine a fixed outcome. As development continues, these early patterns are further shaped by experience, learning, and environment. However, the foundational wiring established during these critical hormonal windows remains an important influence on how the brain processes information and responds to the world throughout life. Fluctuations of estrogen and progesterone across the lifespan play a continuous role in shaping brain function, and unlike early developmental effects, these hormonal changes are dynamic and ongoing. Rather than permanently wiring the brain, they modulate how existing circuits operate from day to day and across different phases of life. This creates a system where brain function is not static, but responsive to internal biological rhythms. One of the most well-studied patterns is the menstrual cycle, during which estrogen and progesterone rise and fall in a predictable sequence. Estrogen typically increases during the first half of the cycle and peaks around ovulation, while progesterone rises during the second half. These shifts are not just reproductive, they have direct effects on the brain, particularly in areas like the hippocampus, prefrontal cortex, and amygdala. Estrogen is strongly associated with enhanced synaptic plasticity, which is the brain's ability to form and strengthen connections between neurons. Higher levels of estrogen have been linked to improved memory formation, increased verbal fluency, and greater cognitive flexibility. This is partly because estrogen promotes the growth of dendritic spines, which are small structures that allow neurons to communicate more effectively. It also influences neurotransmitter systems, increasing the availability of dopamine and serotonin in certain brain regions. Progesterone, on the other hand, tends to have a more calming and stabilizing effect on the nervous system. It interacts with GABAergic systems, which are involved in reducing neural excitability. This can promote relaxation and sleep, but in some individuals, especially when levels fluctuate rapidly, it may also contribute to mood changes or increased sensitivity to stress. The balance between estrogen and progesterone is therefore critical, as it determines how these effects are experienced. These hormonal influences extend beyond cognition into emotional regulation. Changes in estrogen and progesterone can alter how the amygdala responds to emotional stimuli and how effectively the prefrontal cortex regulates those responses. This is why emotional experiences can feel more intense or more manageable at different points in the cycle. It's not just psychological, it reflects real shifts in neural activity and connectivity. Across the lifespan, these effects evolve. During puberty, the rise in sex hormones initiates major changes in brain structure and function, contributing to increased emotional sensitivity and social awareness. During pregnancy, hormone levels increase dramatically, leading to further adaptations in brain circuits related to caregiving and bonding. In menopause, the decline in estrogen can affect memory, mood, and overall brain metabolism, highlighting how dependent certain neural processes are on hormonal support. It's also important to recognize that these fluctuations interact with external factors like stress, sleep, and environment. For example, high stress levels can alter how the brain responds to hormonal changes, sometimes amplifying their effects. Similarly, good sleep and stable routines can help buffer some of the variability. This dynamic hormonal modulation means that the brain is constantly adjusting its function based on internal signals. Rather than being a limitation, this flexibility allows for a wide range of adaptive responses, enabling the brain to shift priorities depending on physiological state. Stress response systems in the brain are tightly linked to both survival and social behavior, and in females, these systems tend to show a stronger integration with emotional and interpersonal context. This does not mean that stress is experienced more intensely across the board, but rather that the brain is more responsive to certain types of cues, particularly those involving relationships, communication, and social evaluation. At the core of the stress response is the interaction between the amygdala, which detects potential threats, the hypothalamus, which initiates hormonal responses, and the prefrontal cortex, which helps regulate and interpret those signals. In females, there is often stronger functional connectivity between these regions, especially between the amygdala and prefrontal cortex. This increased connectivity can enhance the ability to process emotional nuance and social information, but it can also mean that stressors tied to relationships or social environments have a more pronounced impact. Hormones play a key role in shaping this system. Estrogen, for example, can increase the sensitivity of the amygdala to emotional stimuli, particularly those that are socially relevant. It also influences how effectively the prefrontal cortex can regulate that activity. Depending on hormonal state, this can either enhance emotional insight and responsiveness or make it more challenging to downregulate stress responses. This dynamic interaction helps explain why stress sensitivity can vary across time rather than remaining constant. Another important aspect is the involvement of oxytocin, a hormone often associated with bonding and trust. In the context of stress, oxytocin can shift behavior towards seeking connection rather than withdrawal. This has led to the idea that, in addition to the classic fight or flight response, there is also a tend and befriend pattern, where stress increases the drive to connect with others as a way of restoring safety. This response can be highly adaptive in supportive environments, as social connection helps reduce physiological stress markers like cortisol. However, this same system can become a vulnerability under chronic or negative social conditions. If relationships are a source of stress rather than safety, the heightened sensitivity to social cues can lead to prolonged activation of stress pathways. This may contribute to a higher prevalence of conditions like anxiety or depression, particularly when stress is tied to interpersonal dynamics. The brain is essentially amplifying signals that it has learned are important, but in doing so, it can become more reactive when those signals are negative or unpredictable. There are also differences in how stress is processed over time. Females may be more likely to engage in rumination, which involves repeatedly thinking about a stressful event or emotional experience. This is linked to activity in networks involving the prefrontal cortex and default mode network. While this can support deeper processing and reflection, it can also prolong the stress response if not balanced with strategies that shift attention or regulate the body. Importantly, this system is highly adaptable. With supportive environments, effective coping strategies, and regulation practices, the same sensitivity to social and emotional cues can become a strength. It can enhance empathy, communication, and the ability to navigate complex interpersonal situations. This means that the stress response is not simply about how strongly the brain reacts, but about what it prioritizes and how it integrates information. Particularly when that information is tied to relationships and social meaning, neurotransmitter systems such as dopamine and serotonin are central to how the brain regulates mood, motivation, reward, and emotional stability. And in females, these systems are strongly influenced by hormonal states. Rather than operating independently, hormones like estrogen and progesterone interact directly with these chemical systems, altering how signals are transmitted and experienced across different brain regions. Dopamine is closely tied to motivation, reward-seeking behavior, and the ability to experience pleasure. Estrogen has been shown to enhance dopamine activity in key areas like the prefrontal cortex and the striatum. This can increase motivation, focus, and the drive to pursue goals, particularly during phases when estrogen levels are higher. It also affects reward sensitivity, meaning that certain experiences may feel more engaging or satisfying depending on hormonal state. This modulation is not constant, which is why energy levels and motivation can shift over time without any obvious external cause. Serotonin, on the other hand, plays a major role in mood regulation, emotional balance, and overall sense of well-being. Estrogen increases the availability of serotonin by influencing its synthesis, receptor density, and reuptake. This can lead to improved mood stability and a greater sense of emotional resilience when estrogen levels are elevated. Conversely, when estrogen levels drop, such as during certain phases of the menstrual cycle or during menopause, serotonin activity can decrease, which may contribute to mood fluctuations or increased vulnerability to stress. Progesterone adds another layer of complexity. It interacts with neurotransmitter systems in a way that often promotes calming effects, largely through its influence on GABA, the brain's primary inhibitory neurotransmitter. However, fluctuations in progesterone, particularly rapid increases or decreases, can affect how these systems stabilize mood. In some individuals, this can lead to increased sensitivity, irritability, or emotional variability, especially when combined with changes in estrogen. What makes this interaction particularly important is that it creates a shifting neurochemical environment. The same external situation may be experienced differently depending on where someone is in their hormonal cycle, because the underlying neurotransmitter activity is not the same. This is not simply psychological. It reflects real changes in how neurons communicate and how brain circuits process information. These effects also extend to stress and reward processing. Dopamine pathways influence how the brain evaluates effort and reward, while serotonin helps regulate emotional responses to those evaluations. When hormones modulate these systems, they can shift how challenges are perceived, either as manageable and motivating or as overwhelming and discouraging. This can have a direct impact on behavior, decision-making, and emotional responses. It's also important to recognize that these systems are highly sensitive to external factors such as sleep, nutrition, and stress. Poor sleep or chronic stress can disrupt both dopamine and serotonin function, and when combined with hormonal fluctuations, the effects can become more pronounced. On the other hand, stable routines and healthy behaviors can help buffer some of this variability. This dynamic interaction between hormones and neurotransmitters creates a brain environment that is constantly adjusting, allowing for flexibility in mood, motivation, and emotional processing, while also introducing variability that reflects underlying biological rhythms. Brain plasticity refers to the ability of the brain to change its structure and function in response to experience, and this process remains active throughout life. In females, this plasticity is especially dynamic because it is continuously influenced by the interaction between genes, hormones, and the environment. Rather than being fixed, the brain is constantly updating itself based on both internal biological signals and external experiences. At the cellular level, plasticity involves changes in synaptic strength, the formation of new connections, and the pruning of existing ones. Hormones like estrogen play a significant role in this process by promoting synaptic growth and increasing the density of dendritic spines, particularly in regions like the hippocampus and prefrontal cortex. These areas are critical for memory, learning, and decision-making, meaning that hormonal states can directly influence how effectively the brain adapts to new information. What makes this system particularly interesting is that hormonal fluctuations do not just affect momentary brain activity. They can alter the brain's capacity to change. During periods of higher estrogen, the brain may become more receptive to learning and forming new connections. This creates windows of increased adaptability, where experiences, habits, and environmental inputs can have a stronger impact on neural circuitry. At other times, the brain may shift toward consolidation and stability, reinforcing existing patterns rather than creating new ones. Genes provide the underlying blueprint for how this plasticity operates, but they do not dictate fixed outcomes. Instead, they set parameters for how responsive the brain is to change. Environmental factors such as stress, social interaction, education, and physical activity interact with these genetic and hormonal influences to shape the direction of that change. This means that two individuals with similar biological profiles can develop very different patterns of brain function depending on their experiences. Stress is a particularly powerful modulator of plasticity. Acute stress can sometimes enhance learning by increasing alertness and focus, but chronic stress tends to impair plasticity, especially in areas like the hippocampus. Hormones interact with this process in complex ways. For example, estrogen can offer some protective effects against stress-related damage in certain contexts, but this protection is not absolute and can vary depending on timing and overall physiological state. Another important aspect is that plasticity allows for recovery and adaptation. Even if certain patterns of thought or emotional response have been reinforced over time, they are not permanent. With repeated experiences, such as learning new skills, forming supportive relationships, or engaging in regulation practices, the brain can reorganize itself. This is particularly relevant in the context of mental health, where changes in behavior and environment can lead to measurable changes in brain function. This ongoing adaptability also means that the expression of biological differences is not rigid. While genes and hormones influence tendencies, they do not lock the brain into a single way of operating. Instead, they interact with lived experience to produce a wide range of outcomes, allowing for flexibility in how cognition, emotion, and behavior are expressed. Over time, this interplay creates a system that is both biologically grounded and highly responsive, where the brain continues to evolve based on how it is used and the environments it engages with. Over time, this interplay creates a system that is both biologically grounded and highly responsive, where the brain continues to evolve based on how it is used and the environments it engages with. This means that while genes and hormones set important conditions, they do not define limits. They shape possibilities. With consistent input from experience, behavior, and environment, the brain can continue adapting in ways that support resilience, learning, and long-term cognitive and emotional flexibility.